Good afternoon. I am Dr. Lauren Hughes, and I am very excited to share an overview of the work I have been doing to better understand the genetics underlying Pituitary Pars Intermedia Dysfunction in horses. This neurodegenerative disease, also known as equine Cushing syndrome, affects up to 15 to 30% of horses above the age of 15. The disease occurs when a portion of the pituitary gland, located at the base of the brain, overgrows. This overgrowth is due to a lack of dopamine activity and then leads to excessive levels of certain hormones within the horse. These hormones can then contribute to a variety of clinical signs that can significantly affect the health as well as the use of our horses. Some of these include a long, shaggy hair coat, a loss of muscling, changes in behavior, recurrent and chronic infections, and laminitis, a painful condition that affects the hoof, causing loss of use or even sometimes loss of life. A challenge currently facing veterinarians like myself is that this disease can be very difficult to accurately diagnose, especially in its early stages. Interestingly, recent work has revealed that the changes occurring at the tissue level in horses affected with this disease are similar to some of the changes that occur in humans affected with Parkinson's disease, like the famous actor shown, Michael J. Fox. This has presented an exciting opportunity to now better understand the genetics of this condition in horses, as well as to potentially improve the way we screen, diagnose, or even treat these horses. Additionally, the work we are doing may have the ability to translate back to human medicine and increase what we already know about Parkinson's disease. Many diseases are a combination of the genetics an individual has and the effects of their environment. The goal of this project is to focus on and better understand the genetic aspect of PPID in horses by identifying genetic mutations that contribute to an increased risk for development of this disease. To address this goal, we started by obtaining DNA samples from 36 horses, 23 of which were affected by PPID, and 13 that were healthy, normal horses, not affected by the disease. This DNA was then whole genome sequenced to allow identification of mutations or areas within the DNA that are different. Whole genome sequencing is a process that allows us to take a DNA sample from an animal, normally in the form of either hair roots or blood, and determine the entire genetic code. Like is shown in this graphic, the DNA is isolated, broken down into small pieces, and then is assembled in a sequence through a multitude of complex steps. The sequence is then compared against a reference to identify mutations. At this time, we are still in the early and explorative stages of this project, but we have already identified over 130 genes that may contribute to PPID through their role in biologic processes or relationships to Parkinson's disease. Closer evaluation of these genes has revealed over 80,000 mutations, some of which are high or moderate impact. This means they have a larger effect on protein coding and potentially functions of the body. We now are in the process of evaluating each individual mutation. This is a very time-consuming process, but very important because each individual animal, or even all of us as humans, have millions of mutations present within our DNA that may not cause disease. This is why it is so important to actually compare these mutations identified between affected horses and normal horses to better understand their significance and the potential role these mutations play. Since we believe PPID is a complex genetic disease, multiple of these mutations are likely working together to increase the risk of its development. In the coming years, we will continue to expand the number of genes and mutations we are evaluating, and eventually plan to reassess the identified mutations in a larger group of both PPID positive as well as healthy control horses. For many of us, horses play a very important role in our lives. As advanced vet care and management strategies have improved, a larger number of horses are living longer, making this disease more common. Therefore, increasing our knowledge on the genetics underlying PPID has the potential to be extremely impactful to the equine industry. Identification of these potential disease-causing mutations will allow us to develop genetic testing to better screen and diagnose these horses so the treatment can be started earlier before we see the devastating consequences. Another exciting impact of this work may be its ability to cross species and increase the knowledge we have about Parkinson's disease. This work is a collaboration between researchers at the University of Minnesota and Oklahoma State University. Thank you for listening. I hope you enjoyed this video.